So, today we are dealing with one of the most important topics of the whole microbiology, which is actually the testing of the food specimens, which is to ensure the quality of the food, that there is no bacteria inside the packaged food material that are gonna cause highly infective disease in the body of the person who's gonna eat that food. Okay, so now we are going to study the bacteriology of food and food poisoning here. Okay, so first we are gonna study reasons why we are doing all these things. Okay, why we are doing the bacteriological examination of food. Okay, then we will move to the next part, which is testing of quality of food. Okay, so what quality of food is deliverable in the packaged food items, that we are going to study next on the next slide. Okay, so thereafter we will head towards food poisoning as a core main topic, and we will study some of the bacteria which are highly we can see which can cause food poisoning. Okay, so we will study step by step. First of all, we are gonna see some reasons why we are doing bacteriological examination of food. Then we will go to the precautions, some precautions we can take to inhibit those bacterial actions, and from there onwards we will start. Okay, so let's get into it. Reasons why we are doing bacteriological examination of food. Okay, it is to establish standard hygiene by total viable bacterial count of the packaged food. Okay, how many bacteria are there present in the packaged food is important to evaluate, because the type of bacteria present in the packaged food item can be highly infective to the human species who are gonna eat them. Okay, then the second reason is to establish shelf life of the food by detecting food spoilage bacteria. There are some certain food spoilage bacteria which can very quickly degrade the quality of the food which has been packaged. Okay, so to ensure that the food is not gonna be soon not gonna be ruined soon, the food quality is not gonna be disturbed very soon. Okay, so for that we are gonna detect the food spoilage bacteria in that packaged food. Okay, then third reason is to detect specific pathogenic bacteria responsible in an outbreak of the food poisoning. This is the third reason. Okay, so when the patient come with the complaint of like stomach ache and nausea, feeling, vomit, etc., certainly doctor will diagnose it as a food poisoning. Okay, if there is some other sign and systems associated with it, then doctor can initiate a new process of finding out the new cause of this thing. Okay, food poisoning thing. But here the doctor will directly diagnose first for the food poisoning. Okay, when the patient comes with the symptoms like vomit, nausea, doctor will see the food poisoning cause first. Okay, then the doctor will prescribe and bacteriological examination and culture of the of stools, of vomit. Okay, so these kind of specimens can be tested to know which bacteria is causing the food poisoning. Exactly when the bacteria is identified, then the antibiotics are assigned to the patient to eat time to time. Then this effect of the bacteria goes down and bacteria are killed. So then the patient will get relief from the food poisoning. Okay, then some of the precautions that we can look upon, which can easily improve the shelf life of the food and can inhibit bacterial actions. Okay, so as refrigeration is a very good process by which the cycle and the processes and the metabolism going on in the bacterial system, bacterial cell, are slowed down by the refrigeration. By salt and sugar, by salt and sugar, the bacterial cell water actually come out and bacteria dies, and some preservatives are also used as a precautionary measure. So now we will head towards the next one, which is actually the testing of food quality. Okay, testing of food quality. Now we are heading towards heading towards testing of food quality. Okay, so testing of food quality is gonna be tested in the next step. Transport in original pack, must be refrigerated. These are the two conditions which are to be fulfilled while the food sample has been transported. Okay, while transporting the food sample, it must be present in the original pack. Okay, it must be present in the original pack, because if someone ate the food from the original packet and from the one original packet, and for specimen the sample is look, a person ate from shop A. Okay, from shop A, a person took a food from shop A. Okay, so if the person got sick, this sample, from the food sample from this shop, must come to the hospital. Okay, food from this shop should not come to the hospital. Okay, must be original pack and must be refrigerated. Because while taking this packaged food from the shop to the hospital, the bacteria can also die. If the bacteria will die, then what are we gonna diagnose in the hospital? So it must be refrigerated on the way while it's coming from the shop to the hospital. That is why we are taking care of refrigeration here. Okay, so what is the procedure of testing the food quality? Okay, what is the procedure of testing the food quality? Very simple and very standard procedure it is. So let's follow it step by step. Step 1 is aseptically weigh about 10 gram of food. Okay, keep 10 gram of food and weigh aseptically 10 gram of food. Keep 10 gram of food. Okay, second step, homogenize in sterile diluent. Pour some diluent on it. Like this is a bowl, and pour 10 gram of food in it. Pour some water in, pour some diluent, sterile diluent. Sterile means it doesn't have any contamination. Okay, so step 3 is make dilution of food suspension. Make dilutions. Make dilutions means dilute it more. Okay, 10%, 20%, 30% according to the lab, whichever standard procedure it is following. Okay, then step 4, perform colony count, any standard plate count method. Okay, any standard plate count method. Here plate count method can be, there can be a pour plate method, pour plate method. Okay, this is the standard method which is mostly used. So step 5 is inoculate various media, McConkey for enterobacteria, azide blood agar for enterococci, neomycin blood agar for Clostridium tetani, for Clostridia species. Okay, Step 6, identify the isolated bacteria. Identify the isolated bacteria. ठीक है. So this is the thing, okay? Food poisoning by eating contaminated food. How food poisoning is coming to the body, okay? How food poisoning is occurring, okay? By eating contaminated food. Contaminated food means it may contain toxins, it may contain bacteria, live, okay? It can contain live bacteria. If live bacteria is present in the food, then this kind of infection can happen, okay, by salmonella. If the toxins are present in the food, then these can cause the infection, okay? So let's understand first what are these two types of food poisoning, okay? First, in which bacteria directly get into the body and directly infect the body, okay? So in this, the bacteria goes into all the way into the stomach and to the duodenum. From there onwards, it infects the duodenum. Then it gets into the blood circulation. Actually, in severe cases, it also reaches to the brain. In some cases, in severe cases, okay. So it causes typhoid fever, as we know, typhoid. Okay, so this is causing mainly typhoid fever, and this is going itself to the body and to the infected area to cause the infection. But here we have three other species: Staphylococcus aureus, perfringens, and botulinum, Clostridium perfringens, Clostridium, and botulinum. These are the three species of the bacteria which do not go themselves in the body to infect the person, but what they do is very interesting, that they release their toxin when they are growing in the food. Let's take the let's say one example. Let's say this bowl has a food, okay, and this bowl also has a bacteria, some bacterias. Which bacterias? These three bacterias, okay. These three bacterias are on the food in this bowl, okay. This bowl has food and this bowl has three bacterias, these three bacterias, and these three bacterias will grow in this bowl on the food by taking the food as a medium of their growth. So thereafter they will grow in a large quantity, they will grow in a number, and they will grow their colonies in it sometimes, okay. But with the growth, one thing also comes in the game, and which is a villain, which is main villain of the story, which is toxins. They release, my dear friends, they release toxins, and these toxins, trust me, can harm your health to hell, okay. These can harm your health very much, okay. So these release toxins in this bowl, okay. So this bowl has now toxins, okay. So this bowl, I want to just heat this bowl. So if I am a science student, okay, let's say I'm the science student, okay. So what I will do is just pick this bowl up and just put on the induction and heat the induction, so that I will get ensured that all the bacteria of this food in the bowl has been destroyed, okay. As a science student, I will do that, okay. But there is a game. The bacteria will die, but their toxins are still alive. Their toxins are protein, and these toxins will cause harm to your system, okay. So are you getting my point? Okay, so these bacteria are gonna cause a nice and very, very cruel disease. Now we know we have food poisoning, diagnosed with food poisoning. Now the time to know which bacteria is causing the food poisoning, okay. So let's now find which bacteria is going to be mastermind here, okay. So now we will start with these steps. Okay, if a doctor diagnoses food poisoning, if he suspects that it can be a salmonella, okay, a microbiologist will inoculate it on XLD agar, hectone and selenite F broth to see how the salmonella is growing. If it grows on salmonella, then salmonella has been confirmed that it is causing the food poisoning. The source can be man, animal and bird. Okay, so killed on the boiling, it is killed on boiling, it is heat labile, it is not heat stable, it can be killed easily by heating. So mainly it's seen that the food is contaminated after the food is made and after the food is cooked. Okay, so it can also be present in the raw food like undercooked meat, cream, coconut and raw food. Okay, so like the salmonella hates heat. If something is heated and something is properly cooked and your hands are being properly sanitized before eating anything, so you will not get infection of salmonella. That's my guarantee. Okay, so examination, what kind of examination we can do here is homogenize the food in the sterile diluent. Okay, so this is the standard step which is there to be followed in each and every examination of food poisoning. And second is inoculate in XLD agar, xylose, lactose, and the second is hectone agar, and third is selenite F broth. Okay, then the growth, the bacterial growth is to be isolated and just tested that it is salmonella or not. If it is confirmed as salmonella, then accordingly some antibiotics will be prescribed by the microbiologist and the doctor will finally give the medicine. Okay, so we will move to the next, which is Staphylococcus. First is already discussed here, salmonella, and second will be here Staphylococcus bacteria. Now we are on second cause of the second cause of food poisoning, that is Staphylococcus aureus bacteria. So the source can be nose, skin of man, nose and skin of the man. Okay, killed by heat, it doesn't loves heat, it hates heat. Post cooking contamination can happen. Okay, so it is not alive when it is cooked because the heat kills it up. Okay, then the contamination can happen post cooking. Okay, after cooking the food, the food is left open here and there, any kind of infected hand is going in there and touching in there can in fact the food contaminate the food, release enterotoxins in that food. Okay, this is the toxin released by it, enterotoxin. Okay, so specimen can be nasal swab. Pay attention here, specimen can be nasal swab. Culture, it can be cultured on mannitol salt agar. For this, culture was XLD and selenite F broth. Okay, these culture for this, and for this there is mannitol salt agar. Okay, if it grows on mannitol salt agar, then it can be isolated from that culture, and that can be by phage typing of the isolates. We can pinpoint the accuracy that the causative agent is Staphylococcus. Okay, for pinpointing we can do phage typing. Phage typing can actually reveal that bacteria causing the food poisoning is Staphylococcus. From Now we will move to Clostridium perfringens. Now we are on next bacteria, that is Clostridium perfringens. Clostridium perfringens, its source can be animal, meat, and meat when cooked in bulk. Okay, meat cooked and meat when cooked in bulk can be the cause of Clostridium perfringens food poisoning. Okay, spores can survive the heat. You cook, overheat. The proteins of the food will denature, but the spores will survive. Okay, so on cooking the spore germinates. On cooking, look at the chronology. On cooking, spore germinates, and spore on eating it causes diarrhea and produce enterotoxins. Enterotoxins once produced, then it starts kicking the main problem in the body. Specimen can be food and feces, same. The food should be well preserved, refrigerated while transporting, and transportation must be careful, and feces by the patient. Okay, so culture can be direct plating on the neomycin blood agar, and the inoculation on RCM, Robertson cooked meat. Identification and typing must be done, and to identify the toxin, ELISA can be helpful to identify the toxin. Which toxin is causing Clostridium perfringens food poisoning? ELISA can help to identify that toxin. Okay, so now we will move towards the next bacteria, which is botulinum. So I am writing here side by side that which medium is used for its growth. Now we are on the next bacteria which can cause the food poisoning, which is Clostridium botulinum. It is the most dangerous bacteria till third. Okay, we discovered 3 bacteria, and this is the most powerful and dangerous bacteria which can harm directly your brain also. Okay, so it is anaerobe means it can survive without oxygen. Canned food might be a perfect place for it to multiply and survive. Canned food have no oxygen, and it will use all the oxygen will be used by the other bacteria, and it can grow in the anaerobe conditions very nicely in the canned food. Okay, so the source can be soil and animal. Okay, contamination is pre-cooking because while cooking the heat cannot damage the toxin released by this bacteria. Okay, so these toxins are heat stable toxins. Okay, these toxins are not destroyed by heating, so even pre-cooking contamination can happen. Canned food, home preserved meat, veggies and pickles. You see, the box of pickles typically in India have like no oxygen for so many long period. After that it is used to eat finally. Okay, so in that process this bacteria can grow in that medium. Okay, produce preformed powerful neurotoxin. This is my dear friends very dangerous thing, and this can cause neuroparalytic condition. Which is paralysis of brain functions. Okay, so specimen can be food, feces and vomit. Culture can be done on neomycin blood agar and Robertson cooked meat, which are generally anaerobic in nature. in nature. Okay, so these can be used as the medium. So this is, my dear friends, the most harmful bacteria. Okay, so next we will discuss about bacillus. This is the last bacteria we are gonna study today about the food poisoning, which can cause the food poisoning, which is bacillus. Okay, the selective media used to grow if bacillus is infecting your body and causing the food poisoning. It can be isolated from the body and can be grown on phenol red egg yolk polymyxin agar, and can be seen there that this bacteria is doing so or not. Okay, contaminated milk and boiled rice. Okay, it contaminates milk and boiled rice because it is saprophytic in nature. It grows on that, and if that is eaten by anyone, that can be infective. Okay, long time ago cooked rice eating. Okay, काफ़ी दिन के बासी भात पड़े हैं, आपने उठा के खा लिए, तो ये आपको ये इन्फेक्ट कर सकता है. So this is for today, guys. Bacteriological examination of food and food poisoning. Here Saurav Jha signing off.